Yes, now it works. Hi, hello everyone, and welcome again to a live stream. Yes, I can hear myself as well, and it's always good. There's usually a couple of seconds delay, that's why I have to wait a little bit. Hi everyone, welcome again to another live stream, Microscopy live stream. Here's my microphone. Today is Sunday, right? As you all know. Yeah, I had to move the live stream one day later. I want to go back to the Saturday schedule in the in the future again, but I had to move move it to a Sunday to today because I had an appointment yesterday. But I hope and I'm happy that you still found your time today, right? Today I would like to put some food under the microscope because I think last week or 2 weeks ago, someone of you suggested, can you not go into the supermarket and buy things and just put them under the microscope? And I thought that's a cool idea. However, I'm not just going to buy anything. I decided to place a stronger focus today slightly on biotechnology. I do have a few cheeses here, but not only that. I've also got some kefir over there, and I'm going to put some beer under the microscope as well. There's a reason why I would like to do that. Some alcohol-free beer. I prepared something. Would like to talk a little bit about that as well. Yeah, and we're going to see how far it's going to take us. If you have any questions, please feel free to ask them again. However, there's one slight change compared to the previous sessions. I will be trying to answer all of the questions during the live stream. In the past, I've always waited for the off-topic questions for later on. I might still do that, but my son is already in bed because tomorrow is school again, and today it's Sunday. So yeah, and it's quite late already here where I am. So first of all, hello all around the world. I'm just going to generally say hello. Yeah, so I'm not going to read everything now. And people have joined in from all over the place, and it's really cool. Okay. Also, thank you to Alexandre. Yeah. And he posted quite at the beginning. You're such a positive influence on my life. Okay, thank you very much for the nice feedback. Okay, microscopy is also a very important hobby for me. It also keeps me grounded a little bit. And because for those of you who don't know yet, I am a teacher in middle and high school, and sometimes so basically for this reason I also like to do these things here because sometimes I'm also showing the videos to my students for some simple lab things. So what I'm going to do, however, for right now is the following: is I'm going to put the cheese away because there is fungus on here that I would like to show you. However, there isn't one thing that I would like to show you first, and I'm going to carefully put this away. Because what I've done is I prepared, and I'm going to show you a short video of what I prepared. I centrifuged some beer. Okay, I generally don't drink beer, but I bought myself a bottle of alcohol-free beer, and it's a beer which was cloudy. So there are some beers out there which basically still contain some suspended particles, for example, yeast. And I decided, well, I'd like to know a little bit what is in the cloudy beer because on the label it said that it contains yeast. And I think I was not able to find yeast, but I'm going to quickly show you the video first that I made. And then we're going to look at this sample here. So I already today prepared a little bit. So here is the video. So this is, you can already see over here, that's the beer. I'm going to pause a little bit here because the beer obviously, let me remove the arrow. The beer obviously contains carbon dioxide. It's bubbly and foamy, and I let it stand for a couple of hours for all of the gas, the carbon dioxide to disappear. Otherwise, you're not able to centrifuge it. And you can see that the beer is quite cloudy. So it's not, so there is some suspended particles in there, and I wanted to figure out what is it. For those of you who speak German, it's called a Weizenbier. And here I've balanced it because for the centrifuge, you have to make sure that everything's balanced, otherwise there are vibrations. Yeah, so I used it on a scale and simply used a pipette and put it into my centrifuge. So that's basically one beer. That's a balance, and I had 2 tubes, right? And I've got 1 tube here. Then I turn it on for about 5 minutes or so at maximum speed. I think 4,500 rotations per minute. And and it's basically a couple of minutes later, I took it out, and something that's important now is it looks much more clear and a little bit difficult to see, but on the bottom part here, here I'm going to pause again. I can actually point with the arrow here. You can actually see that there is a little bit of grayish stuff in here and also here on the side. It's not easily visible here in the video, but that's called the pellet. And this means that the suspended particles, whatever was in the beer, now collected down here on the bottom, and that's what I would like to put under the microscope. Okay, so let's move on here. Where's the? I have to medias. Yeah, I've got to click play again. Yeah, so that's essentially what I have also here. Here as well. So I made 2 tubes. One of them I looked already today in the afternoon, my time, right? And the other one I've got here. And this pellet is sometimes very sensitive, so this means if you shake the tube, of course, then everything's going to go into suspension again, so you have to carefully remove. It's a little bit blurry, it's a little bit out of focus, but I carefully remove the so-called the supernatant of the beer, and I put it in there. So that's here. This is the original beer, again, without CO2 because I let it stand. And you see, if you look, it's kind of turbid, it's cloudy, it's not quite clear, because it's the type of beer, and the clear one, the supernatant, is so basically the thing that I just removed. You see, it's much more clear, right? So essentially, this is a method of concentrating. And what I would like to do now is I'm going to take so we're starting again. So what I would like to do is I would like to take this pellet in here, resuspend it, and put it on the microscope slide, and then we're going to look at it. That's it, right? And a little bit to my disappointment, I was not able to see nice yeast cells. This was kind of a little bit my hope. And for reference, I've got some yeast, baker's yeast here as well, which I would like to put under the microscope so that we just see how it looks like, okay? But this actually shows that beer is a natural product because it contains so many other things as well, right? So, so what I'm going to do is I'm going to quickly pull up and down, and I'm resuspending this pellet. Okay, so there's only a very small drop of beer left here, and I'm just going to resuspend everything carefully. I think that should be enough already. Yeah, and I'm going to take it up, and it goes on the microscope slide, of course. So don't be disappointed, okay? It's not this is not one of the most exciting specimens, okay? I just wanna pre-warn you, if you expect something really cool or so, then you're probably gonna be disappointed. But it's still interesting to see a little bit of what we're able to see here, because for me, the take-home, not the take-home, it's a surprise, was I expected some kind of, I don't know, some kind of nice large yeast cells, which I was not able to find, right? So here it is. I'm going to switch over to the square microscope, not square, I labeled it to my microscope. And here it is. I need to turn on the light. Okay. The arrow I'm going to take away. If you see, look, if you see those fibers here, okay, that does not mean that it's in the beer, okay? It could have been, it's most likely contamination from a microscope slide and so on, right? So don't worry about these things here. Don't place a stronger importance on those contaminants. So let's go up with the magnification here a little bit. We see an air bubble, yeah? And you see tiny little dots, yeah? And it's basically what we see here. And yeah, again, this is could be from the tissue paper, so not to place a too strong an emphasis on here. 40 times, and that's what we see here. And when I saw this, I said, that's super disappointing, because normally what you have is when you have this cloudiness, usually that is because they left the yeast inside the beer. But then I was kind of thinking a little bit, and I said, well, actually, maybe it kind of makes sense a little bit, because you see, if the yeast were still in there, then it would settle to the bottom of the bottle, because the yeast is actually heavier much denser. And what you see here is only very tiny particles that are still suspended, because beer, of course, is a natural product. So you see all sorts of things in here which I cannot even identify. Let's go up over to 60 times. Let me also adjust the filter accordingly. Yeah. And you're also going to see that there is a little bit of movement going on. And that is because of the so-called the Brownian motion. I mean, some of you probably already have heard about that before if you watched my previous videos. It's because of the random motion of particles what we have here. Okay, so this is basically a little bit the unexpected thing here. And what I then did a little research, and it could be that these are indeed yeast particles or fragments of the yeast cells, or it could be that the beer is slightly filtered. Because if yeast were in the beer to a significant amount, it would probably also settle down, and it wouldn't be cloudy, but actually it would settle down to the bottom. Okay, so every now and then I'm going to just have a look in here. If you, somebody wants to pineapple juice. Gee, exactly, I forgot. Okay. The pineapple juice as well, fruit juices generally, because fruit juices contain, of course, fruit cells. Yeah, and I'm just reading through the comments here. Is alcohol-free beer just beer with the alcohol removed, or is it made completely differently? I think what I heard once, I'm not an expert on this, you can use yeast that produces low concentrations of alcohol, and then it is removed later as well. I read somewhere that some kind of like reverse osmosis can also be used to I guess there must be some kind of techniques here as well. Yeah. It says here, if too many yeasts are left in there, it can restart fermentation too, and then basically when you open it, the whole thing blows up, right? Because the yeast continues. Especially, I could imagine if it's you see, if there is alcohol in the bottle, then the alcohol would inhibit the fermentation process. But if it's alcohol free and it contains yeast, then of course I can imagine that fermentation would kind of go on, and maybe even alcohol would be formed. Right, and then it's not alcohol-free anymore. So this is kind of an interesting thing. But on the label of the beer bottle, it actually said it's cloudy because of the yeast. But then again, it wouldn't, as we mentioned now, it wouldn't make a lot of sense to have yeast in there, because then fermentation would continue. Then you would have alcohol again. Yeah. So that is So that is the thing that pineapple juice particularly interesting due to the microscopic needles. Some crystals evidently or something. Yeah. So so both is possible. The beer can be diluted to less than 0.5% alcohol, or beer can be heated till the alcohol evaporates. Yep. Yeah. So I guess there must be several possibilities here. In any case, I think it's kind of shows how still complex it is. I know it's not the most exciting specimen here, but I just wanted to show it to you in any case. What I would like to do now is before I show you the other things, I would like to go here a little bit, and I don't know, many of you probably have already seen yeast cells before. But I'm going to show you in any case. For whatever strange reason, I've got problems these days obtaining some fresh yeast. They used to sell them in little packs, square packs, but mostly now yeast is sold dry. I suppose because maybe people are baking less and they don't need less of the fresh yeast, and maybe it also for storage purposes. Yeah. So what I simply do is I simply resuspend some of this. Also something for the future, what I've been experimenting as well, is of course if you add sugar to the whole thing here, and if you wait a couple of hours, you're going to see that the yeast starts dividing. It's called the budding process, and you can also observe this under the microscope. Yeah. So again, I'm going to take a very small amount here. So I'm going to cover glass on top, and then we should be able to see how this looks like. Okay, so this is a very little liquid now, so we should get a very flat specimen, which is nice. And let's go again here. Okay, air bubble. Okay, I mean I did not give it a lot of time to actually suspend, but I mean you get the idea. This is much larger, 40 times. Yeah, this is much larger and much more uniform, 60 times. Yep, here we go. Yeah. So yeah, and occasionally what you're able to see, no, maybe not here so much, but sometimes you can actually see that one of the yeast cells contains a little, like a little outgrowth, a little bit. That's called a bud, and that's a form of that's cell division, mitosis, but in yeast it's asymmetric. So you have basically, asymmetric cell division, and it's called budding. And if you add a little bit of sugar to it, and if you wait, then you can actually see that, a small bud will grow out, and then later on, maybe this one over here, I don't know, yeah. And then later on, you will see that actually the bud will also start to increase in size. Yeah. But again, this here is dry yeast, so it probably takes a couple of minutes for it to become active. Yeah. So I'm going to go through the whole thing. What is yeast? Actually, it's a fungus. Yes, it is related to the fungi. It's a non-filamentous fungus. Yeah, so it's a eukaryote in that sense. It has a nucleus, and it belongs to the fungi. Yeah? So today, basically, I'll be showing you a whole bunch of fungi as well. Yeah? So this basically is is a fungus, obviously. Huh? So I'll go back here about the pineapple. I don't think that they're crystals. They seem to be microscopic spines, which I believe is why eating it makes your mouth tingle. Okay, good. Yeah. I don't know what they are. We could have a look at it, and maybe if it's polarizing, it could be that the little spikes in it. Maybe the could be an indicator that it might be a crystal. I don't know. Yeah, yeah. So that's basically how yeast looks like, and that's what I expected, and I did not get that in the beer. Oh, this is embarrassing again. Okay, maybe. Okay, what in the world is this here? How many of you recognize this? This is it looks very much like a starch grain. What in the world does a starch grain have to do in here? I think I did not clean I'm recycling my microscope slides, right? And maybe I didn't wash it properly. Yeah, so this looks very much like a potato starch grain here. Yeah, it's also polarizing. So always wash your slides properly. So, but yeah, I don't know. It could be. Yeah. So that's basically yeast. Here I'm using my 60 times objective. Okay. Yeah. So yeah, I'm every now and then when I'm pausing, don't worry. I'm just trying to read the comments here. Okay. So this is basically this was basically the yeast thing. So what I would like to do now is I'm going to go back to the desk here. Okay, and let's have a look at because yeast is a fungus. Let's have a look at at some of the cheese fungi as well. In case I want to do the following. I also want to give you a very short overview of how my table looks like here. It's a little bit messy, so I'm going to flip the camera over so that you get a better orientation. Here, this is basically where my monitor is, right? Here, that's the camera on top. We're filming me, and what I have over here on the side. I mean, you cannot see this now. Here, I collect all of my used slides in this plastic container. I'm washing them later. Yeah, and this over here is some yogurt. So maybe we're able to see some yogurt bacteria as well. Yeah, and I put the monitor on this platform over here, so because I need the space here on the bottom as well. Yeah, so just that you see a little bit how this whole place here looks like. So this basically then goes into the plastic container. So the yeast I'm finished with, and I would like to now give it a try. Let's put the tomato away. I would like to give it a try now with the cheese here, and this is fresh cheese which I bought yesterday. And why cheese? Obviously because it contains a fungus, because sometimes people want to see fungi, right? And you want to have a safe source, right? And that's of course a safe source. And yeah, a little bit of biology first. So basically, what's going on here? The fungus that you have on cheese is from a culture. That's important. You do not just allow the milk to become rotten. You have to add, or they are adding a specific culture. And that's really important because you want to make sure that the fungus does not produce any toxins or poisons or anything, right? Otherwise, you can become quite ill. But there are certain cultures that are known and essentially that are reproduced deliberately for the purpose of making cheese, and especially for the camembert, what we have over here. I just recently read that it's been so optimized from cheese making that it does not make any spores anymore. Yeah, so apparently some cheese makers in some countries are a little bit worried now that it will become more and more difficult to kind of continue breeding it because it's very difficult to get it to make spores. Yeah, so because it's such an optimized culture, yeah, fungal culture. So what we do now is the following. I'm just going to again take I don't know, I'm just going to put this here. I have to take a little bit of this white. The name of the fungus is Penicillium. Now, if you think that why in the world does it sound like penicillin, Penicillium? Well, that is because indeed penicillin, the antibiotic, was discovered in 1927 by Alexander Fleming. He was a Scottish researcher. By accident because a fungus from the genus Penicillium made a substance which was able to kill bacteria. And that's why the first antibiotic was called penicillin. Yeah, so it's basically the penicillin is named according to the fungus. Yeah? So I'll add a little bit of water now. So I prepared also a little bit of amount of water, and we're gonna put this here. And now I have to kind of resuspend it a little bit and break apart the clusters of cells. And otherwise it might be too difficult to see. And some months ago, I think I made a video, if I remember, and I put some camembert fungus under the microscope, and indeed, I was not able to see any spores. I don't know, maybe it wouldn't be good anyway if there were too many spores here, because then it would kind of spread over all over the place, and then they're going to settle down on food, and then they're going to start making more fungus. Don't forget one thing, that those fungi that you use for making food are only safe when they actually grow on the food that they're meant to grow on. It could be that if the fungus now grows on a different type of food, that it will start to produce certain poisons, okay, toxins and so on. Yeah, so it's not good. Yeah, so I don't know if this is sufficiently done well. In any case, I will now put a cover glass on top again, and let's have a look. Let me see here. Let's have a look how well this works. Sometimes I'm catching two cover glasses, and then again I'm going to read a little bit from the comments section, the scope view. I have to always turn up the, yep, here we go. I mean, it's now the 10x, that's where I've taken it. Again, and those round things are bubbles, which shouldn't worry us too much. Some contaminants maybe. You see, it's definitely a contaminant from somewhere. Yeah, and what we're able to see now, go up yet one more further. Yeah, and that's called the what I always used to say hyphae, but I was corrected. It's called, you're supposed to pronounce it hyphae. The hyphae are the cells of a fungus. Yeah, and that's basically the fuzzy thing here. And normally, when I put a fungus under the microscope, I see a lot of spores, but not here. Okay, and this kind of shows a little bit that indeed for whatever reason the Penicillium fungus doesn't like producing spores. Okay, so I'll just Aha! I have to laugh. I'm reading the comments. Aha! I now see the pineapple needle in my sample. So apparently some of you are, putting stuff under the microscope while you're watching the live stream. Yeah? Yeah. Physical comparison change of cooked food. Yes, it's also possible. For example, some food, if you cook it, especially the starch, will change it quite a bit. Yeah. What would be the water to yeast sugar ratio? Let me quickly check here. I mean, the point is the following. I guess what happened if you would like to put some sugar into the yeast. There is a how shall I put it? If you add sugar to yeast, then it will start forming CO2, right? And you can see the CO2 bubbles. If you add too much sugar to the yeast, then it's not going to do that anymore because of the high sugar concentration. I do not know what the ideal yeast sugar ratio is to actually see the budding of the yeast. I can only tell you the following, that one of my students wants to now do an experiment of actually testing different types of sugars, whether they can actually induce the budding faster than other types of sugars. So this is actually an experiment that I'm doing with one of my students, where we'll be adding different types of sugars at different concentrations to yeast, and then observing them at a regular interval to see how many % of the yeast actually starts to bud. I only have read somewhere that if the sugar concentration is too high, then fermentation will also stop. But again, fermentation and the cell division of yeast, the budding, is actually something different. Yeah. So I have to tell you, sorry, I won't be. I don't know what the ratio is, and I guess it also depends on the sugar. On the type of sugar, but I remember that if you Google a little bit, it's such a common thing to observe the budding of the yeast, and folks who are into fermentation and so on, they actually study this stuff. But I don't know it off the top of my head. Yeah. So basically here, that's the camembert. Just for the fun of it, let's put it into phase contrast. Last week, I talked a little bit about phase contrast as well. And phase contrast, I only have a 40 times phase contrast objective, has the advantage that you're able to see structures of very low contrast. So you now see that the individual cells of the camembert are now darker over here, for example. How does this look in bright field? Because I've got so-called DIC. This is now regular bright field. And this here is now DIC with the color. You can play around with the color a little bit as well. So, but that is basically, I think for the other one over here, it's a different fungus. It might be, a little bit different. So we're going to have a look. I'm going to throw away the toothpick because I do not want to have a cross-contamination. The cheese I'm going to eat later. Okay. Every now and then I'm going to again pause and have a look. I hope I'm not seeing anything. Yes. Okay. Paul is saying because those cells are called hyphae, and I always being native in German, I always say Hyphae. So, just wrote my Abitur exam on Friday, age of 36. Both parts were ecology. Okay, not genetic. Okay, which was odd. Okay. For cheap do-it-yourself wine, it's about 250 grams white sugar for 1 liter of water and 3 grams of dry yeast. Okay. Thank you for the info. So if you want to make wine, you add 250 grams of white sugar to 1 liter of water and 3 grams of yeast. Okay. 1 teaspoon of sugar for 5 teaspoons of water. Okay. Thank you very much. That's interesting. So I'll try this as well, but you have to be a little bit patient. I guess temperature also plays, of course, an important role here. So back to the desk. Okay. I like talking so much that I'm getting a little bit carried away here because I still do want to show you a few things here. I do also have a banana here, an avocado, a mango, which I'd also like to have a look. So I'm going to quickly. I also have to change the tip, and again some water. Maybe I just put down the water first. So let's have a look here. This here, oh, you don't see it? Yeah, here we go. Yeah, that's yeah, there's some blue mold in here. So you always have to be a little careful because it's easy to also take some of the cheese along, yeah. And but I would like to have it, if possible, as yeah. Maybe yeah, I don't know. I'm going to put this a little to the side here. Let's see how well this works. And I have to tell you, I mean, I studied microbiology a long time ago, but still, keeping those cultures pure and everything clean, that's a challenge, I think. Yeah. So yeah, you always, I guess, you have to make frequent tests that your culture doesn't become contaminated, and so on and so on. So let's have a look. I hope I got enough of the sample here. Okay, here we go. Okay, wow. Lots of other stuff here as well. Lots of particles, probably some milk protein. I'm a little those fibers, I don't know. You have to be careful. Sometimes people say, "Oh my gosh, I found something in my food. It's this fiber," but it doesn't have to be necessarily in the food. It could be something actually on the microscope slide. So let's have a look here. I think I might have found something. Okay, here it is. Here is something. Okay. And what do we see? What do we see? Maybe you also realize. Okay, I see a lot of dots, which are quite bright, which basically means that they're highly refractive. So it could be that maybe these dots that you see here could be spores. I don't know, right? But I don't know if these, or maybe some, I don't know. Sometimes it could be some fat droplets, but fat droplets actually are more strongly visible, and sometimes they have different size as well. So I'm kind of interested a little bit in those dots here, if they could be spores or not. But this, what you see over here, yeah, these are definitely the cells of a fungus. Yeah, so that is. So it was, by the way, a long time ago when I was finishing university, I had this final oral exam. It's a commissional exam. And one of the questions that I was asked was actually, how, if you see something under the microscope, how do you actually know if you're looking at a fungus just by looking at it, or at bacteria? And one of the criteria to distinguish is that fungi generally are branched. At least the filamentous fungi are branched. Bacteria generally don't do that. But you see that over here, yeah, it's kind of, it branches off. You know what? Let's go up a little bit with the magnification it further. Okay, let's open it, the condenser, yeah. So that could be, yeah, and yeah, I don't know, could this be fat droplets? I mean, one has to be always a little bit careful that one doesn't jump to too early conclusions, yeah. But yeah, could be, yeah. That these maybe are some kind of from the milk fat or something like that, yeah, and not necessarily spores, yeah, because they do have slightly different sizes, yeah. So yeah, one has to be a little careful. I don't know, yeah. So every now and then I will have a look here. Let me see. So make a solution: 1 teaspoon. The needles in the pineapple juice are due to calcium oxalate crystals known as raphides. Okay. Would these dissolve in alcohol? Not sure. Solubility in water is okay. Late to the party from the UK. Okay, hello. Just Googled, very interesting. They do cause what I thought they did. I didn't know that they were actually a crystal though, which is really cool. Yeah. Okay, no, calcium oxalate can, by the way, also if you put some onion, the dry onion skin, not the one that you find between the layers, the fresh one, but this brownish onion skin, try putting that under the microscope and you're also going to find calcium oxalate crystals. They look quite nice as well. Yeah, so it's a dry, papery onion skin. That is also, they also contain quite nice calcium oxalate crystals. Okay, so yeah, so that's basically the other type of fungus that we have here. Yeah, and here yeah, you see that. Yeah, let me have a look. Actually, maybe my sample that I caught wasn't the best one. Yeah. Sometimes it's also like this that in some cheeses you have actually a complex assortment not only of fungi but also of bacteria. Yeah? But those long structures that you see here, those filaments, these are of course the cells of the fungus. Yeah, quite nice. Yeah, it's essentially cheese and so on. That's a form of, if you want to call it like that, some kind of a controlled spoilage. Yeah. And as a matter of fact, you can use bacteria and fungi, like we are using here, to actually make the food store longer. Because if the food is inhabited now by these fungi, then and maybe they're producing certain chemicals that inhibit the growth of others, then actually it is a way of preserving the food. So, yeah. So again, this would be actually quite something interesting. A little bit is to figure out what these circles are. If these are just fat globules, I don't know. I mean, then again, on the other hand, they're quite similar in size. Yeah. One would have to probably, I'm not gonna do that now, but what I would like to do then maybe, I'd go up to the 100 times oil immersion objective and then see if I'm able to see certain structures inside those. Because if there are certain structures visible inside those round things here, then it actually means that they're a cell. Yeah. Again, the motion that you see, mostly Brownian motion. Yeah. Yeah. So, yeah. Where are the nuclei in these fungal slides from cheese? Okay, the nuclei, if you want to, sometimes difficult to see, but let me find some fungal cells. The thing is the following. There are certain fungi out there that contain actually in a cell multiple nuclei. So let's have a look here, right? If you look in, I bumped it. If you look into those here, then you can actually see that in many cases there are structures visible inside the cell. Yeah, and these are cell organelles, including the nuclei. In order to actually identify the nucleus, I'd have to probably do some DNA staining. Okay, but yeah, but you can see that sometimes there are indeed things inside the cell, and this is another indicator that we're not talking about bacteria here. Bacteria, first of all, would be way smaller, and bacteria do not have any cell organelles in them. So if you see cells with an internal structure, then this basically means that cell organelles are present. This looks like a little bit like a conidium here. Conidia are also the spores of a fungus here. Yeah. It's interesting, yeah. So in any case, you see a little bit the difficulty as well when you take a sample from a natural environment like cheese, for example, then there are so many other particles in there as well that it's becoming difficult to actually identify now the real thing. That's one of the reasons why early microscopists, I think even with tuberculosis or so, with certain diseases, it was very difficult for them to actually see which one are the bacteria, because there were when you take a sample from a human being, there were so many cells from the human being also present that it was very difficult to see the bacteria. They were kind of covered up. So the early microscopists, they developed staining techniques that selectively stained only the bacteria, so that they kind of were able to be much better visible. Yeah? So, starch can be identified using iodine staining. Is there a way to identify fat globules? Yes, there is a stain called Nile Red, just like the YouTube channel. And this Nile Red apparently stains hydrophobic substances like fat and also plastics, apparently, because they're also hydrophobic. I've not used that stain, but indeed that would be a possibility. Yeah. So there are stains that actually do that. Yeah. There is a YouTube channel also called Nile Red. It's a chemistry YouTube channel that I can highly recommend as well. So last but not least, and then I want to move on, because concerning starch, I do have a banana here that I would like to do some iodine staining as well. Okay. Originally, I actually wanted to make some permanent slides, but because there are some time constraints as well, a little bit. What time is it? Yeah, 40, it's fine. So I'm going to put the slides away, and I'm going to try the next one. Okay. So let's try to take something from here, from the green part. Let's see if I don't know if I'm able. I got something here. Okay, some you don't need a lot. Why the fungus? Well, the fungi are so-called saprotrophs, to use an ecological term. And what they do is, I know it sounds a little bit yeah, unique when I say that, but the fungus will release enzymes into the milk, into the cheese, so to say, to digest the cheese, right? And the digested cheese are then the nutrients for the fungus. So if I were to leave this now here for a couple of days, you're going to see that the fungus would actually slowly grow and cover everything here, even that surface. Yeah, and this kind of gives the fungus its typical, the cheese its typical taste. Yeah, because of the enzymes that the fungus have released. So it's yeah, when we're eating cheese, or other milk products, where it's kind of like we're eating pre-digested food. And as a matter of fact, that's the whole point. Huh? So let's have another look here. Stereo, nope, that's the wrong microscope. Here we go. Okay, it's again a little too bright. Let me see. This is a little... maybe I don't know. Again, we're seeing those. Huh. Now they look way more red. I don't know. Now these look like spores now somehow. So maybe we actually found some spores finally. Okay. Yeah, this is actually a much nicer sample. I don't know. This looks, I don't know. The fat droplets would be much more refractive. The contrast would be much larger. So I'm quite sure that these are actually, yes, they are. Look what I found here. Look here. Very nice. Yeah. Here you actually see how the spores are forming. Yeah, yeah, that's part of the cells, and here at the end you have the spores, yeah, being given off. Yeah, quite nice that we're able to see that here. Yeah, so these are definitely spores. Yeah. The nice thing, no, not nice. I'll tell you what I tried to do, and it was not successful yet. I've taken spores from a fungus, and I wanted to actually see how they grow on a microscope slide. So adding a little bit of water and sugar, but you see that's very difficult because if the conditions are not right, they're not gonna germinate or grow. And that would be kind of nice somehow if I were able to take those spores, put them on a microscope slide, add the correct nutrients and whatever, and actually watch them germinate and grow. You can do that also with pollen, but you have to add also the right concentration of sugar, everything that's all very delicate. Yeah. It doesn't mean that these spores like to grow in the water either. I mean, they need a surface to grow on, right? The fungi, yeah? And so this can actually mean that it's difficult maybe to get them to germinate. But that would be kind of nice. Huh? So again, I'm going to have a short break. Not break, I'm just going to quickly read the comments again. Okay. We might not test it yet, be able to stain the fat globule with fluorescent dye from a highlighter pen. I can imagine, never tried it. Whatever the dye is, if the fluorescent dye must be hydrophobic. So it must then stick to the oil. Yeah. You could do it with aspergillus, but it's bad for your health. Ooh, yeah. Yeah. Huh? Don't use the word stick. Don't use the wood stick. I use metal. Okay. Yeah. What I've basically, I guess you're referring to the toothpicks here, right? Yeah. What I'm doing is, I use clean toothpicks because I later on want to eat the cheese. So that's the reason. So okay, I'm going to move on because I want to show you something. Something which I might not put under the microscope. I mean, I can. No, I tried it, but it's difficult to put under the microscope. But I'm going to show you in any case. Where is this? And some of you might know this already. I'm going to put this away. Look what I got here. It's not yogurt. Okay. You open. It's of course milk is in here, and I need a fresh spoon, and I've got some fresh clean water here as well. Okay, because it is food, right? So, and if you want to eat it, then and this is in case you've never seen this before. This is I'm making something called kefir. It's also it's not yogurt. It's also a fermented milk product, but you see, it's slowly starting to become solid. But I have to find the kefir fungus because there is a fungus in here, which I want to show you. Okay, and so here it is. And basically, what you do is you when it's finished, you pour it through a sieve, you catch the fungus, and then you put it into fresh milk. I'm going to rinse it now in this water here. Okay, this is just This here is just regular fresh water, and I just want to show you how this. You don't see it here. This is how the fungus looks like. Yeah, it looks almost a little bit like a coral. Let me see if I can focus this here. Yeah, and it's rubbery, and it's not only a fungus, but actually it's a complex aggregate of fungi and bacteria. And it's kefir, and basically it will grow, it will increase in size as it grows, and it will ferment the milk. And then you have a milk drink which is similar to yogurt, tastes slightly acidic as well. It's basically the fungus that's responsible. I tried to put this under the microscope once, but I was not able to see anything properly because I was not able to get it thin enough because it's very thick and rubbery. Yeah. So again, this is because there is, you said, Alexandra is saying, great demonstration for the yogurt. Strictly speaking, per definition, it's not yogurt. Okay, it's kefir. And this is a form of so-called milk kefir, which basically is for milk, but they also have water kefir, where you can make some drinks. And some of you might have already heard of kombucha before, where you actually add tea. Yeah. So it's it looks so gross. I feel like I've produced similar masses when I've had the flu. Yeah. So this is actually quite. I consider this quite interesting, and there are different types out there, and and they will grow, and then when it basically becomes too large, then it will kind of split, and it will separate. And what I can, however, do is just because it is a microscopy channel, I'm going to let's see if there's anything in here. Yeah, no, this stuff here not. Yeah, see, yeah, maybe it's fine. So you know what I'm gonna do? I'm gonna put this away. And then later on, I'm going to put it back into the milk until the milk fermentation is finished. But let's do the following. Let's put it under my stereo microscope. I have not looked at it under the stereo microscope before. Okay, so I'm just saying that. You don't see my face. Where am I? Here I am. Haha. Hi. Okay. So let's have a look here. I have to reach over. Yeah, and this is basically how it looks like. Yeah, and that's why does it look so, I don't know, like a cauliflower almost, like, yeah. And the reason is, of course, it's always the same story: to increase the surface area. All right. If it were just smooth, then there would not be not all of the parts would be in contact with the milk. But by having this irregular shape, of course, then the different parts are better in contact with the milk. And look, it's a spoon right now. Let me go down with the. No, that's already the lowest magnification that I have. Yeah, and it's really like almost, it's almost rubbery, you know. It's quite fascinating. I mean, let me just a second. Let me try the following. I mean, I could try to put one thing under the microscope, but it's, yeah, I got this tiny piece here. Let me try it in any case, but it's not, honestly, I think it's not, we're not able to see a lot, okay? The reason is because it's so tight, so tightly packed that maybe we're not able to see individual cells here. I tried this once and I was not really successful. But just for the fun of it, we're going to try it in any case, because that's a little bit the how do you say this, the point of this channel, to simply try out things even if they don't always work. Yeah. Add a little bit of water always. No, I don't know. I don't remember. Yeah, I don't. This was, see. Yeah, see how am I supposed to get this flat? It doesn't. Let me break it apart a little bit. So I'm gonna give it a try, but we're probably only going to see a black blob, yeah, a shadowy black blob, because it doesn't allow the light to go through. But who cares? We're just gonna try it in any case. Where is it? Here we go. Yeah, yeah, yeah. Maybe some cells broke off. I don't know. No. We'd have to look a little bit, yeah, between here. Nah, you see, a bubble. So, I mean, what I would normally do now is, if I'm seeing those bubbles, is wait for an hour or so. It's because sometimes if it starts to make CO2 and ferments, then essentially those bubbles start to grow. But I think you get the idea. There's not really a lot to be seen here. It's too thick. Yeah, it's actually worse than I expected. Yeah, yeah, yeah. Forget it. I think they're more interesting. Yeah. Um, I don't know if these are cells there or not. Let me see. Also difficult to distinguish. Yeah, these do look like a little bit like cells. In any case, yeah, this is. I'd have to probably take a little more time in picking it apart and getting it very thin, yeah, and then that would be possible, yeah. Is this a single fungus or a colony? Well, it is a colony, but it's. I think it's not. Somehow, colony kind of implies a little bit like that. It's just one type of fungus. But from what I've read and researched, is that especially kefir is a complex association of fungi and bacteria and so on. So it's not a pure, how do you say, individual organism, so to say, but it's actually a combination of yeah, I suppose maybe even symbiotic organisms that kind of work together, right? So in that sense, yeah, containing fungi, and I think the wouldn't be surprised, but usually when something is very thick and sticky, usually it's some kind of a polysaccharide that kind of holds everything together, right? Yeah. But that's essentially, I would say, almost a whole ecosystem that we have here. So what I'm going to do is I'm just going to put this back again. And what one could do is one could pick it out every day and then measure how heavy it is. So I'm just going to put it back, and maybe it's going to yeah, continue its fermentation process. Yeah, so that's basically one thing here. So it's another type of fungus. And what time is it? Yeah, I will knocked over the things here. So this here is some yogurt. Also, something that I would like to say, I did not make that, but normally if you want to see bacteria, nice bacteria, they look really great, but not in commercially bought yogurt, but in homemade yogurt. And the reason is that the commercial yogurt, very often they have mixed it, yeah, and this kind of has broken apart those nice chains of bacteria that you normally would see. If you make it yourself at home, you boil some milk, you cool it down, and then you add a spoon of yogurt, and then you wait for a day. If you make the yogurt, I've seen some very nice bacteria, yeah? Still in chains, but normally when you look at them, yeah, from commercial yogurt and everything's already been mixed. And what we have here as well is that, well, I didn't catch anything, that sometimes we see, of course, a lot of milk particles as well. So this goes over here. And tardigrades are living in my apartment. How is that possible? I have moss, but I haven't taken time to watch them yet. Haha, yes. Moss is some moss contains a lot of tardigrade, I found, and some moss not at all. So if you're not successful finding tardigrades in moss, then change the moss sample. You're going to save time this way. The reason is that because if tardigrades are present, I sometimes found there are a lot of them present, right? And so let's have a look here. So and let's have a look here again. These are now scope. Here we go. Again, a lot of probably milk protein. And here, what we have to do now here is we have to look between those gaps that we see here. We go up, and yes. So if you want to see some nice bacteria, what you do is you take a small amount of yogurt. Okay, a very small amount of yogurt. And here we go, very nice. And these here are bacteria, and some of them still are a little bit in chains because we're lucky. If you look carefully, then you can see that some of them are actually in short change. Okay, also over here. And if you make yogurt yourself at home, and if you don't shake it, then you can actually, I've seen some huge changes. Yeah, so that they look quite nice. And that's actually because many people want to see bacteria, right? To start off with microscopy, they want to see bacteria. Where can I find bacteria? And I'm saying, you don't want to grow your own bacteria because you don't know if they're safe or not. But here with yogurt, they are safe, right? Because of the yogurt bacteria. See also here chains. Yeah, these are all cocci. This basically means they're round. And because this is DIC in bright field, it looks like this. Okay. That's how you would see them. And bacteria are those specimens that you want to observe also in phase contrast, of course. So I have to change everything again to open it. And this is how it looks like in phase contrast. Where I have to refocus. Interesting, because normally the bacteria appear darker on the white background, but here it's actually different. Let me open here. Yeah, here now it's darker. Okay, you see that this is now only 40 with a 40 times objective, so it's smaller. But you see that they appear now darker also over here on a brighter background. But I think even in bright field it looks kind of better. Okay, so let me switch it back. Okay, so that's the next, So you see that this is basically in bright field again, biotechnology. I've knocked over again the toothpicks. Okay. Yeah. And what you see over here on the side are usually some milk proteins, milk fat, of course also bacteria, but you have to look at the liquid and it's basically where you see them most easily. Yeah. Yeah, I watched spiral salmonella from chicken. Yeah, you got to be careful with the salmonella, of course. Yeah, of course. Yeah. Okay, I'm just reading again some of the comments here. How bad does kefir milk smell? Where did I put it? It does smell. Yeah. Honestly, this one does not smell particularly pleasant. I wonder why. I wonder if I've got some contaminants in there. I have to tell you the following, that the kefir that I've got here, I had in my refrigerator for a couple of months. Okay? And then today or yesterday rather, I exchanged with new milk. So maybe it will take a couple of generations or days to regenerate. So, but honestly, I don't like the smell of this one here. So if it smells kind of bad, this doesn't smell like, then probably there's something else in here as well. Yeah, that shouldn't be in there. So what I could do is I could try to, you don't, yeah, you got to be careful with microorganisms in any case, right? And if it's something that doesn't smell very good, then you don't want to drink it. Yeah. So, uh, so I'll quickly read every now and then something here. Tardigrades, tardigrades, tardigrades. So, okay, so let's move on. What time is it? It's already 1 hour. So I'm going to now switch a little bit. We've looked at fungus, we've looked at the kefir, you looked at the yogurt. A few things I did not look at yet, which I would like to look at, is the following. Well, I did look some months ago at this. Look at this banana here. This is what happens. This banana is not rotten, okay? But this is too extremely ripe, and when you keep it in the fridge, it turns brown. So, and it's extremely soft, which is nice, because when it's very soft, you can put it easier on a microscope slide. So look at this, yeah? So it's very soft, very ripe, but it's not, excuse me, it's not, yeah, bad in the sense. I'm taking a small sample here, and now I'm going to do the following. I'm not going to put it directly on the microscope slide, but look what I have here. Still have a little cough. This is iodine, okay, and I diluted it a little bit because when you work with yeah, I have some concentrated iodine, but I'm a little bit reluctant of using concentrated iodine because when you spill it, it's kind of very aggressive, right? So for safety reasons, yeah. So it might take a little longer for the iodine to stain the starch. Yeah, and as we're waiting here, I'm going to read again a couple of comments here. Okay. Who saw the fake microscopy video of the banana with a nematode? Okay, I have to ask the creator to make real micro content instead. There are a couple of, there are a whole bunch of microscopy channels out there that are more or less serious. But I've seen some channels where, oh my gosh, come on, that's not how you prepare. They take I don't know. They take the whole banana. I'm not making this up. They take the whole banana and squeeze it on the slide and put it under the microscope. And then they kind of show something. And I said, what's the point? Why even bother making videos if you don't want to show it the proper way? I don't understand. Yeah. They make more views. Yes, that's one of the comments. That's sad, I think. Yeah, I agree with you. But I don't know if you've seen this YouTube short channel. I could not believe this. It says, yeah, I mean, they took a whole object, they just squeezed it directly on the slide with they pressed it totally wrong, and they put it under the microscope and they show something. Well, actually, you couldn't see anything anyway. But so why even bother making content like this if yeah, I don't know. So we can see that it starts to, at least some parts starts to turn dark a little bit, and these are the parts I would like to look at. Of course, if I you see, the point is the following: concentrated iodine is quite corrosive, right? And therefore, I do not want to accidentally get the iodine in contact with my microscope objectives or so, and things like this can happen. So what I'm going to do now, therefore, is I'm going to rinse it a little bit in water as well. Yeah, so I'm going to take it out. You see, it didn't stain all the way. A part of it is a little bit darker. And I'm just going to quickly rinse it. Yeah, and that's enough. And I go over here. And now I have to pick it apart. I'll take the darker darker part, and I will make a so-called this one I have to squeeze now. But let's put this away. Let's put this back. Who cares? And this one I have to now squeeze to get it flat. And here I have a slight problem because this surface here is padded. So if I press too hard, then I might break the slide. I have to press carefully. Yeah, you don't want to move it horizontally, but you still see that some parts are darker than others. And now let's have a look at this, and let's see if we are able to see the starch grains of the banana. So again, this works only with a very ripe banana. That's why I've taken this one here, scope. And where is it? Here we go again. Okay. So let's have a look here. Yeah. So over here, very nice. Let's go higher magnification. This is not bright field, by the way, right? And yeah. What we have here, all of those, okay, obviously the round things that you see here are air bubbles, right? And all of those dark patches, these are the starch grains. Some of them are clustered together quite in, so let's find some of them where they're a little bit more separate. Maybe over here. Yeah, here we go, right? Yeah, you can see here, you've got a cell in here, and then inside the cell you've got those starch grains. Yeah. That's basically the reason why bananas give us so much energy, because there is plenty of starch in there. Here as well. You can of course see them also without staining them with iodine. Yeah. So that's not a problem, but with iodine it simply looks much brighter, much more contrasty. Yeah. These are air bubbles. Okay. So I've seen some at the wrong place. Oh, interesting. Here I don't seem to see them. Sometimes in some bananas I could see them quite easily, even though they were not stained. Yeah. Yeah. So, but you get the idea, right? Yeah. So iodine being obviously a very popular stain here. Plenty of starch inside the banana, and the iodine evidently did not reach all of the parts. That's why not everything's uniformly stained. Do these starch grains start as vacuoles? Ooh, I don't know. I mean, obviously, you do need enzymes to make them, and usually those starch grains are crystalline in structure as well. So I don't know how they are actually made. That's actually a good point. Yeah, if they are actually starting off inside some kind of vacuoles, where then there are these enzymes present, where essentially more and more glucose molecules are connected together until you've got a starch grain, because starch grains are, of course, made of long chains of glucose. No, sorry, I don't know. Here we've got some unstained starch grains. Here, that's what I wanted to find. Yeah. So you see that you are able to see them without. So let's let's go up with the magnification again a little bit here. Here they are. Yeah. Here they are. They're kind of still clustered together quite a bit. Yeah. Let's see if let me see. Look, they seem to be also a little bit polarizing as well. Obviously, because they're so dense and compact that they're crystalline, and then they're also a little bit polarizing. Yeah. But we do it like this. Let's do the following. I've never looked at them using my phase contrast. So let's have a look how they look like with phase contrast, if you actually see something here. No, wrong filter. Yeah. Well, actually, okay. Interesting. It actually looks quite okay. In phase. Yeah. Okay. Yeah. Those long things that you see in bananas, sometimes you also see that they're spiral. These might be some vascular tissue transporting liquids. Yeah. Yeah. What's the spiral structure to the left? Where is this? Is this xylem? That's xylem, yeah, of course. Yes, they are. Okay, that's it. That's the xylem. So yes, so what you see over here, that's actually it was not in focus. You in plants, you often see spiral structures. Actually, it's not even that clear here, and these are basically the vessels, the cells that transport water in a plant, right? And those spiral structures basically are thickenings of the cell wall to prevent the vessel from collapsing. Those, yeah, I've seen actually nicer ones before than this one here, but it's definitely xylem. Yeah, let me see. Yes, here it's better. Now it's better a little bit. Yeah, yeah. Here we go. I've seen ones in a banana. They were huge. Yeah, extremely long. Yeah. They can either be spirals or they can be rings, separate rings, and different plants have different structures here. Yeah. Yes. From the ketchup special. Yes, some months ago I put ketchup under the microscope, and I was also able to find those spiral structures. Of course, because tomatoes also contain xylem, right? I found them also in pumpkin. You find them all over the place. It's yeah, in yeah. But this one here is yeah. Okay. So let me quickly check again. Yeah. So yeah, the ketchup, the ketchup special. I put some ketchup under the microscope, and I think some, if I remember correctly, I took some. Here's another one. Some ketchup that was a little bit older already, and I found a lot of some fungi as well, and I don't know what. Yeah. So because once it's open, the ketchup, then of course you have bacteria and so on, fungi growing in it. Yeah, if the contaminants. That's why you have to, once opened, you should eat the food fairly quickly. So that's what time is it? Yeah, I do still want to put the thing here because I'm not going to eat it because it's already too ripe. I never put look at this. What I got here. So, and essentially the reason we forgot about this. This was in our fridge, and it doesn't look very nice anymore. It's a mango, and probably not good anymore. But it's so soft that essentially, let's have a look. Yeah, it's already brownish a little bit. Yeah, so and it smells. It smells a little bit already fermented and like alcohol. Huh? So, but why not? Why not put this a little bit under the microscope? Because I've never done this before. I'm just curious. Yeah, and it's so soft. It is so soft that essentially I should have no problems squeezing it again under the microscope on the slide. It's way too much. I just realized this. It's huge, this piece. And yeah, I'm going to. This is not gonna work. Here we go. That's better. So let's put this on here and let's see if I'm able to compress it. Look, it came out. Yeah, this is not ideal. No, it's too up. Yes, here we go. When you do this compression specimen, never move it horizontally. So, and let's have a look. Okay. What is this? Okay, no, it's fine. So, number one, number two, where is the scope? Okay. These are bubbles. I don't know. Still too thick here. The spiral structures again. Look, here we go. Some xylem again. Here they are again. But it's still a little too thick. I don't know. So the individual cells I'm not able to see properly. It's just a mess. But at least we have found again a little bit of xylem tissue here. Yeah, so that's the mango. So you see that since mangoes and bananas, yeah, it's quite similar. I don't know. I don't know. It is not so. I mean, the yellow stuff is could be some kind of pigment in here, but the individual cells are not nicely visible. Yeah, it's just probably also too thick. Look at this stuff here. I don't know. Maybe some cellular debris that was squeezed out. I don't know. Yeah. Let me have a look a little bit. Yeah. Um, I've learned the word xylem on this channel. Okay. Yes. Yeah. I started. I'm going to. Yeah. It was. I'm just reading the comments here. Yeah, elephants eating a lot of overripe fruit, they got drunk. Yeah. It happens to birds as well who are eating too many berries and then it starts to ferment and then the birds actually have a problem. Yeah. Yeah. So, yeah. So this is, but you know what? Where's this? I'm just gonna try this just for this fun of it. I'm going to quickly put maybe I don't know if this works. Okay, let's take this little mango piece over here. Let's put it also in here into the, I don't know if this actually works or not. This here is the banana. Let's take this out here. Yeah. And let's take it away. And I don't know if the mango actually will, yeah, if the iodine will stay in the mango or not. But you know what? Give me a second. So here it is. This is the original iodine bottle. This is concentrated stuff, okay? So maybe if starch is present, but I can imagine, and that's what I assume, especially because the fruit is overripe, it could be that the starch has already broken down and was used to ferment the whole thing. So I can imagine that maybe there's not so much starch left over here. Okay. So, yeah. But actually, I don't see it turn black. Otherwise, because this is not pretty concentrated, there's no point putting it under the microscope if it doesn't turn black. It did turn a little bit. Let me see. Now I'm a little bit curious myself. You see, I've got over here, I've got the metal tweezers, but because iodine is so corrosive, I don't really want to use metal tweezers on the iodine. So that's the reason why maybe I'm a little too cautious. I don't know, but this should not be a problem now because I already rinsed it. Yeah, I don't know. I'm not convinced, especially this is going to be difficult here to compress. Yeah, and nah, here it is. You see, it's not very nice looking. Squeezed it out here. So I'm going to see. Maybe we're. I don't know. I really don't know if there are any starch grains in here or not. So, scope view. No, I don't think so. Ear bubbles. There are some black dots there, but I don't know. Yeah. No. Yeah, I think this is if the fruit is overripe, then what's happened sometimes. What I read somewhere is that the starch grains are essentially broken down, converted to sugar, and then this is again further broken down and fermented. So yeah, nothing black over here. So probably no starch. I'm gonna put it away because I want to have the iodine away from the objectives. Okay. Dilute iodine won't attack your tweezer. Okay. Yeah. Yeah. That's why it's stainless steel anyway. Yeah. But that's why I diluted it down a little bit. Yeah. I'm reading again. I was the one that suggested the supermarket. Sadly, I had a math test. Okay, no, the live stream will be available in any case. And what I've done today is I've placed a stronger focus mostly on biotechnology stuff like fungi and bacteria. But actually, in an upcoming live stream, I also want to put some vegetables, for example, celery and so on. We can make some cross sections of the stem because they kind of look nice. So the plants I have deliberately, kept away. Yeah, I mean, I do also have a tomato here, but I think I'm just going to wait with that. I'm going to do a plants one. So what I'm going to do now is this. Sorry, yeah, in a couple of minutes, I'm going to call it a day again. A day, that's nice. It's already quite late in the evening. I'm just checking. Okay. Yeah, I'm just reading some of the comments here. A genius idea. Start your short with fake video, food and worm, and then explain the reality and how to debunk a fake. Yes, just for your info, I've received a couple of years ago already. Some folks sent me an email because they were really worried because of some YouTube channel that said, "Oh my gosh, there are worms," and we found worms everywhere. And then I looked at the video and says, "These are not worms; these are just dust fibers." That's not a worm. This is not how worms look like, right? But people actually got worried about that. So where I'm kind of a little bit. Why do you make videos where you're spreading fake news? I don't understand. So and that's something that where I'm getting genuine emails from people that were really concerned. They said, "Oh my gosh, I heard, I saw this in the video that there are basically worms somewhere," and then I said, "No, these are not worms." Huh? I mean, for example, if you have some kind of, I don't know, very often. I mean, I mentioned this already. Very often when you have how do you say, dust fibers. I mean, I've got a lot of dust here on my table. Yeah, you know what? I'm just gonna collect a little bit. Yeah. I'm gonna show you. Let me quickly. Let's see if I was able to pick up something here. I don't know, but people who have never seen worms before, they think that these are worms. Let me see if I caught some dust here. Yeah, and where is this? I don't know if I'm lucky enough or not. So apparently my desk was cleaner than than I anticipated. Yeah, exactly here. This one here, right? Yeah. Some people have seen this, right? And they say, "Oh my gosh, look! If you look careful, it's even moving." Do you actually see that it's slightly moving? You see this? Yeah. Let's go up a little bit with the magnification. Oh my gosh. Right, and it's moving. So I found a moving worm and they're all over the place, right? No, you know why they're moving? It's because they're absorbing the moisture of the water, right? And so they're stretching and So basically, there are lots of videos out there that kind of say, oh my gosh, these are worms or whatever, and no, they're not. Yeah. So yeah. So this debunking idea is actually something that it would be a good idea indeed. Yeah. Yeah. Um, Yeah, clickbait view, and then once they're there, you get educated. That's something. Yeah, I found worms on my banana. I found worms in the food and whatever, right? And then essentially you kind of get the people to click, and then you debunk it, and then you give them proper information. Yeah, I don't know. Yeah, uh, so. Let me quickly go through the whole thing here again. By the way, if any of you have any suggestions of what you want to look at, please feel free to comment and say. I'm always happy for suggestions. Yeah. So what time is it anyway? It's already 1 hour and 25 minutes. So it's time to go to bed, for me at least, because tomorrow in the morning I have to go. Of course, like many of us, to work again. Starch, fat, and sugar are chemicals for organisms to store energy, if I understand correctly. Are there any other chemicals used for that? Let's put it this way. Interesting question. Starch, fat, lipids, so basically starch, polysaccharides, or carbohydrates rather. Carbohydrates, lipids, and sugars and carbohydrates belong together, and also proteins. Okay, so you've got carbohydrates, to which starch and sugars belong to, then lipids, and also proteins. These are the macromolecules, and they are organic, and therefore they contain energy, right? However, different substances have a different energy density. So for example, if for 1 gram of fat, lipid contains approximately twice as much energy as 1 gram of carbohydrate or 1 gram of protein. Yeah, so this is the reason why many animals use fat for long-term energy storage, because fats, lipids are less oxidized. I don't want to get too chemical here right now, and therefore they have a high energy density. So with very little mass, you can store a lot of energy in fats and oils. Yeah. So to answer your question, yeah. Proteins can also be used, but not generally for, I mean, being organic, they store energy, but that's not the primary source that organisms use. So usually carbohydrates and lipids. And I think what I'm going to quickly do here, soil science. Yes, I would like to see more about staining techniques. Okay, thank you. I'm gonna get myself a couple of stains then. Methylene blue I've used a lot. Some stains I'm a little bit reluctant of using, because not all stains are healthy, because stains do react with biological samples. I mean, that's the whole point of a stain, right? And therefore one has to be a little bit careful with some of them, because they might make you ill. Seriously, Microbehunter, you should consider clickbaiting. Try it and see if it's working or not. Try something that generates a lot of comments. You know what? Maybe I'm gonna try this. I'm currently in this channel. Do the following. Maybe some of you are mostly only watching the live streams, but I'm also, of course, uploading regular videos. I've recently changed the format around a little bit. I used to always put myself into the circle, into the corner. I've now changed the format. I'd like to also hear what you think of that. I'm also going a little bit stronger with YouTube Shorts, because more people watch YouTube Shorts. And this way I can also reach a wider audience. So have a look also at the videos besides the live stream here in this channel. And I'm happy for some feedback as well. Okay. Maybe also a session on human secretions, mucus. Yes, that's possible. The thing is, proper explanation. Lots of human cells. If you take sample from your mouth, you'll find plenty of cells. Quite interesting as well. Yeah. Have you ever tried testing a sample of the mystery fog particulates, or would be interesting to see? I'd have to do a little more research about this mystery fog. basically particulate matter from the air. One would have to somehow catch that a little bit, yeah. Either using some kind of sticky tape if it sticks, yeah, yeah. Soil science, fluorescence, yeah, fluorescence. Honestly, I've been working on this a little bit. It is possible, maybe with UV ultraviolet LEDs, to do some near fluorescence using algae. And you do not need fluorescent stains, but it's called autofluorescence, and sometimes chloroplasts and so on, they shine in a red light when you shine UV light on them, or blue light. But the algae have to be treated. They have to be kept in the dark for about 20 minutes. So there is a little bit of preparation necessary as well. And then you need long exposure times because the autofluorescence is very weak. But this is actually also possible. Okay. Okay. Then, yeah, Nile red as a stain. I'll have to see where I can get it. Okay. Yeah. Yeah. And fluorescence. I don't watch the eyebrows video and friendly parasites. Yeah. Okay, basically the eyebrow videos, that's the one with the face mite, right? They don't do anything bad. Yeah. So what I'm going to do now, folks, is I'm going to, I wish you still a nice Sunday, if you, depending on where you are in the world, right? If you still have a Sunday in front of you, but for me, it's already the end of the day, and I'm going to also call it quits for the day. I wish you all the best. Yeah, I hope to see you again next week, and yeah, I'll think of another topic again. And of course, I'm happy for any comments and suggestions of what you want to look at. I'm going to say bye-bye now. Yeah, happy microbe hunting as always, and all the best. Okay, bye-bye.